Okay, so this is kind of the IA guide that you guys have at the end of your notes. It should be the last thing that's in your notes that you got. And so what this does is it really goes through an introduction. It tells you all the different parts. It gives you the rubric and tells you how you're going to be graded. And then it goes through and it breaks down each part and sort of tells you what you should be including in that section to get your credit, to get your full marks. Okay. And so I mentioned last time that this is part of your IB grade. And in fact, it tells you right here, it's actually worth 20%. So your tests that you take in May are 80%. And then they average that with this. This counts as 20%. Okay. So, this sometimes this can help you get a passing grade. If you do really well on your internal assessment, your IA, then sometimes it helps offset if you don't do very well on one part of your test, and it might help you bring up your grade overall a little bit for your IB exam credit. And it's graded on a rubric, and the rubric has 24 marks. So marks are kind of like points for IB. And so it's one scientific investigation. Says it should take about 10 hours of class time. Okay. So that means that you should spend 10 hours total, like doing your research in the beginning, doing your planning and writing out your investigation, and then actually coming in and collecting data, and then doing your calculations and your data processing, and then writing your actual report. And so it says that your report should be 6 to 12 pages long, with a maximum word count of about 2,000. And I will tell you that if it's over 12 pages, they penalize you in the communication section, because they believe that you should be able to get across your information and your analysis in 12 pages or less. So if it's shorter than 6 or longer than 12, a lot of times they will penalize you in the communication section that we're going to talk about later. And so basically, I'm going to kind of scroll down here. Here's the breakdown. These are the 5 different sections. So they grade you on personal engagement. So that's like, it's not like you just did some random lab that like anybody could do. It's that you have some sort of connection or some sort of like curiosity or interest in why you chose what you chose to investigate. The exploration is really your design side. So did you design a good experiment? Did you collect good data? Are all your data tables look nice? You collected appropriate data. Your procedure's easy to follow. Your lab equipment is correct. Like you didn't pick bad lab equipment and things like that. And you collect enough data. So I mentioned last time that you have to choose whatever you choose. You're going to change your variable 5 times. And then for each one, you have to take 3 trials. So you have to do 15 different data collections, 3 for each of your 5 variations of your variable. So if you do less than that, then you'll lose points in the exploration section because you're not meeting that minimum criteria for the data collection. Analysis is going to be things like your how is your data presented? So do your tables all have labels? Did they have the units and the uncertainties included? Are all your data recorded to the same precision, the same decimal places? Are they neat and easy to read? Do you have a graph that graphs your data and shows a line with a trend possibly that you can analyze? And then the actual analysis in terms of showing your calculations, propagating the error and the uncertainty with the calculation, presenting the data that's processed after the calculations, and then discussing it and comparing it to if there's a literature value that you can use and you can calculate percent error, calculating that and discussing it. And then also relating it back to the research, the background research that you should have at the beginning, because you should do some background research so that you know a little bit about the background of the chemical technique and what you're doing and the substances that you're using. So when you get to the analysis section and you're analyzing your data, you can refer to your background information. And, if something came out and it made sense, then you can say, well, based on the background research, it makes sense because of whatever. Or if your data doesn't match that, then you could discuss, well, why didn't you match what you thought you should have gotten? And then that kind of directly leads into the evaluation where you're going to evaluate your data. You're going to, like I said, calculate percent error and look at your percent error and see if you have systematic error. And then you're going to talk about how you can improve your lab. So if you have systematic error, what possibly could have caused that? And then what could you do to fix that? If you were to redo the experiment in the future, what could you fix in your design or your data collection process in order to fix that? And also, could you extend it? Is there anything you could do differently? Do you want to test something, that's related but not exactly the same? And that kind of stuff. And then communication is kind of overall the paper. So like I said, they're going to look at the length. They'll look at like your grammar, your spelling, if you have your citations, because you have to do background research and information. And so you'll have to have citations for any references that you use. And then if you're following sort of the format and the pattern that I talk about in here. So altogether, that's 24 marks. Okay, so this is kind of the breakdown for how it is graded. And then after this, it's going to go into detail. So I mentioned that personal engagement says here it assesses the extent to which the student engages with the exploration and makes it their own. So it can be recognized in different attributes and skills. So it could be a personal interest. Okay, so maybe there's something you're interested in and you find a way to design an experiment that's related to chemistry that relates to something you're interested in. Evidence of independent thinking. So you're thinking outside the box. You're not just doing some kind of what some people call cookie cutter labs that, everybody does them all the time. You're thinking about it in a new, different way. And that's kind of the creativity and initiative in designing as well. And so it doesn't necessarily have to be like, oh, I was interested in this and so I chose this, but you have to demonstrate that you are thinking kind of beyond the norm. You're getting a little creative, you're taking initiative, and you're designing something that maybe somebody else hasn't done it before, or maybe they haven't thought about it quite that way. So here's what the rubric looks like that I have to grade you on. And so you notice that the personal engagement was worth 2. So I can give you 2 marks, I can give you 1 mark, or I can give you no marks, okay, depending on how you do. And so to know which one you're gonna fall into, if you read the statements, they kind of match up. Like on this one, it says, the evidence of personal engagement with the exploration is clear with significant independent thinking, okay, and initiative or creativity. Up here, it says it is limited with little independent thinking, initiative, or creativity. So they're talking about the same thing, but if you want 2 marks, it's going to be clear and significant. If you get 1 mark, it's like you tried, but it's limited. And if it's not even demonstrated at all, then you get no marks, okay? The second part on this one, it says the justification given for choosing the research question and or the topic under the investigation demonstrates personal significance, interest, or curiosity. On this one, it says it does not demonstrate those things, okay? And then the last one on here, it says there is evidence of personal input and initiative in designing, implementation, or presentation. And this one, it says there's little evidence, okay? So that's kind of how these work. Is the one that's full marks is going to be, you're going to get very clear, very high stuff. If you get less than that, it's going to kind of go down until it's 0, okay? So here, and after each section, it's going to have some tips. So some tips, your topic should have personal significance. You should be able to show personal input in designing it, and your outcome should be unknown. You should not pick something that that you know the answer to. The point of investigating it is that you shouldn't know the answer, and you're going to try to interpret your data in order to answer the question that you have, your research question. Now you're going to base it on some sort of chemical theory stuff, so like you could do calorimetry or titration or the rate of reaction. So you're going to base it off of some chemical knowledge, but you should not know the exact answer or the exact outcome for the data that you should get from the experiment. All right, exploration. So this one's worth up to 6 marks. So you can see the way the ranges fall on this one is there's a 5-6 range, there's a 3-4 range, there is a 1-2 range, and then there is 0. So on this one, it says exploration, the criterion assesses the extent to which the student establishes the scientific context for the work. So that's doing your background research before you even start designing your lab. You've done background research and you include that in your report at the beginning because you're talking about, you know, whatever technique or whatever chemical scientific context is going to apply to your lab. You state a clear and focused research question, and you use concepts and techniques appropriate to the diploma program level. So the concepts and techniques for chemistry. So like we've talked about, you know, the different calorimetry ones, the different titrations, the rate of reaction. So all those kind of techniques, that lab techniques that would be something related to chemistry for IB diploma. And then it says, where appropriate, it also assesses awareness of safety, environmental, and ethical considerations. So in your write-up, you should address, if you have chemicals, you want to make sure that you talk about handling them safely. So that, you know, do they need to be stored? Do you need to make sure you have goggles and gloves on? Are you using heat? Because you want to make sure you're careful of heat sources. And then also environmental and ethical. So how are you going to dispose of chemicals and things like that? And so the way that the breakdown goes, it's really similar. The statements are going to look similar. There's going to be a few words that are different. So like the first one on the 5-6, it says the topic of the investigation is identified and a relevant and fully focused research question is clearly described. The difference up here is it says the topic of the investigation is identified and a relevant but not fully focused research question is described. So the difference is that you have fully focused and clearly described. If you go down then to the 1-2 level, it says the topic of the investigation is identified and a research question of some relevance is stated, but it's not focused. Okay. So that's, and I'm not going to go through every single one, but they're all kind of like that. Each statement, it kind of goes down in what the description of it is. So here are some tips, okay? The background information, it should describe and explain the theory related to your topic and specifically your research question. So if you're choosing something related to calorimetry, your background information should have to do with like heat, with enthalpy, with calorimetry, you know, or with the substances that you're investigating with your research question. If you're choosing titrations, then your background information would probably include some information about, you know, acids, bases, stoichiometry, solutions, all that kind of stuff. And it could vary, okay? But based on what you pick, your background information should explain the chemical theories behind whatever you're picking for your research question. For the research question, you need to be specific. It's one sentence, it's a question. It should be worded like this: how does the, and then you insert whatever change you're going to do, your independent variable, affect the dependent variable. It's that simple, okay? So once you determine what you want to test, you could say like, how does the temperature affect, you know, the rate of reaction of, you know, whatever substance? You could say, how does the mass affect this? How does the concentration affect this? How does, you know, blah, blah, something affect something, okay? So it'll change depending on what you pick for your specific topic and research question, but this format is what it should look like. How does the independent variable affect the dependent variable, okay? So your variables, your independent variable, that's what you are changing. And you have to, like I said, you have to do at least 5 changes of it, okay? So let's say that I want to test how does the temperature affect the rate of reaction between 2 substances. I'm not going to pick any right now. But that means my independent variable would be temperature, because I'm going to heat them up or cool them down. I'm going to pick 5 different temperatures to do them at, okay? The dependent variable is what and how you're measuring. So in mine, if I'm doing how does the temperature affect the rate of reaction of some chemical reaction substances, my dependent variable would be the rate of reaction. So I'm going to be measuring time and using time to do rate, okay? The control variables, these are everything that's going to stay the same. So you have one independent variable, you have one dependent variable, and everything else should be a control variable, okay? So what's staying the same is, if in my example for, you know, temperature, rate of reaction, those are my independent and dependent variables. Well, my control variables, I should be using the same masses, the same volumes of substances. I should be doing everything at the same, you know, standard temperature and pressure, because I'm going to be doing it here in the classroom at school. I'm going to do them for I'm going to measure them for, you know, I'm sure I'm going to use the same equipment. Sorry, I lost my train of thought for a second. I'm going to use the same equipment. So I'm going to use the same timer. I'm going to use the same balance to measure all the masses. I'm going to use the same glassware, okay? And there are several other things, but everything that's not independent or dependent, everything else is a control variable. You should keep everything else the same, because if you don't keep everything else the same, then that's not following proper experimental design. Because if we want to make sure that our independent and dependent variable are the only things that are changing, we got to make sure everything else stays the same, okay? You want to state, as it says here, state precisely in the method how you're going to vary the independent variable. So in my example, if I'm saying, you know, how does temperature affect the rate of reaction of something, then I want to state, I'm going to measure these temperatures. I'm going to measure it at, you know, 10 degrees Celsius, 20, 30, 40, and 50 degrees Celsius. So some of them I'm going to have to heat up. Some of them I'm going to have to cool down. Okay. Or if you picked concentrations, you need to state what are the 5 different concentrations you're going to test. Okay. You know, whatever you pick, you need to state clearly how are you going to vary it. What are those 5 different variances you're going to have of it. And then you also need to clearly state how you're going to measure or observe the dependent variable. So, you know, in mine, if I'm talking about how does it affect the rate of reaction, I need to clearly state that I'm going to have a stopwatch and I'm going to be timing, you know, how long it takes for something to happen, how long it takes for the, you know, bubbles to go away and the reaction to look like it's done, how long it takes for something to change color, how long it takes for the temperature change to happen. Like, whatever you're doing, it's going to depend on what you choose. So here's an example. You could do it in a table like this. You could be very specific for all your control variables, because you want to state how you're controlling all the other variables. So, oops. So for example, if I want to control the temperature, then I need to say something like, I want to record the temperature at regular intervals. That way I'm keeping monitoring on it so that I know it's not changing. Okay. All right. So this goes through methods for collecting data. And so, like I mentioned earlier, you want to make sure you're collecting sufficient relevant data. So we talked about that you have to have 5 different variations of your variable, and you have to do each one 3 times minimum. Okay. And so that goes to the precision. We want to collect precise data. That's why we're doing everything at least 3 times so that we can take an average, because that's just good experimental design. It's minimum 3. Really, you should do more if you have time. You want to be specific. If you're using specific equipment, you need to tell me, like, what size beakers are you using, what size flasks, what size graduated cylinders or burettes, are you using hot plates, Bunsen burners, and what sizes are they? And also, if you're using solutions, you need to give me concentrations. So for example, if I'm going to use sodium hydroxide, a solution, I need to tell me the molarity. What's the molarity of it? What's the concentration? If I'm using a solid, I need to say, well, it's zinc powder, and I need to give the mass. How much of it am I using? Okay. I want to describe the details. So if I'm using a volumetric pipette, I want to tell what size I'm using, but I also want to include the uncertainty. That's part of the details of your equipment, your apparatus. Okay. If you're using a hot plate or a power supply or an electronic balance, okay, I want to make sure that I include uncertainties, and I want to include any relevant information about the equipment. Okay. A detailed diagram, that, you know, you don't always have to do that, but it does help a lot of times. I'll take this off now that somebody's gone. Okay. So, and if you can't draw, you can also insert a photo. So you can actually, once you have your equipment set up, you can take a photograph, and then you can put the photograph in your write-up. And then if you want to add labels with text, you can do that. But in some way to have visuals, because visuals are usually always helpful. Okay. It helps when you're describing something to show somebody a picture of what it should look like with labels of all the different equipment or the substances. And then it says, describe a method that will allow you or another person to carry out your experiment. So when I say describe a method, like, you're going to do steps, like you're going to do, like, number 1, do this, number 2, do this. Okay. So you should be telling me steps, not like a paragraph description of your procedure or your method. Okay. Once you've given me all the background and you've given me all the equipment and the substances and all that stuff. You need to tell me how many trials you're going to do. I already told you that minimum you're doing 3 trials per thing. If you have time and you want to do more than 3, you are welcome to, but the bare minimum you have to do is 3, okay? And usually doing 3 of them 5 times, meaning you have to do 15, that usually takes people a pretty long time, okay? Especially because you guys are usually new to doing this, and so it takes you time to learn, you know, because in the beginning you're gonna make mistakes the first couple times and you might have to redo something, okay? And that kind of takes a little bit more time. All right, into the analysis section, it says this is the extent to which the student's report provides evidence that the student has selected, recorded, processed, and interpreted data in ways that are relevant to the research question and can support a conclusion. So that's kind of multiple parts here. So it has to be relevant to your research question. So always go back to your research question as what you're doing relevant to that. And you're supporting a conclusion, okay? So you collected enough data and you processed the data, you did calculations, you made a graph, you did all kinds of whatever, well enough that you can support a conclusion that addresses your research question, okay, and tries to answer it. So again, here are your descriptors. So same kind of thing. This one says the report includes sufficient, relevant, quantitative and qualitative raw data. So raw data is like what you directly collect from your measurements and your observations, and it's in a data table, okay? Appropriate and sufficient data processing is carried out with the accuracy required to support or enable a conclusion. So data processing, that's where like you're taking the raw data and you're doing something with it. So you're doing calculations for something. You're taking it and you're making a graph of the data so that you can interpret the data on a graph, okay? The report shows evidence of full and appropriate consideration of impact of measurement uncertainty. So that means that all of your errors are propagated. So all those uncertainties, they're propagated throughout calculations, and you include uncertainties on all of your numbers and measurements and calculations, and also on graphs, okay? The process data is correctly interpreted, that a completely valid and detailed conclusion can be deduced. So they're just basically saying that you've done enough with the data to actually have a valid conclusion that addresses your research question. And then as you go down, obviously the statements look similar, but it's going to be like, you didn't do it as well, it's not clear, it's partially valid, those kinds of things. So tips for analysis, okay? You want to record all relevant data and observations in a table, okay? We looked at that in the topic 11.1 stuff last week. The data table should be organized. You should have headings with telling you what you're measuring and the units and the uncertainties for the piece of equipment. All of your measurements, the quantitative data should have appropriate units. So quantitative, maybe you're measuring temperature, masses, volumes, pressure, absorbance, pH, time, okay? It could be a lot of other things too. But they have relevant units and uncertainties. For qualitative observations, usually you might want to do that before and after. So like if you're doing a titration, it says the final color at the endpoint was a faint pink. If you're doing something that says during the reaction, the bromine water changed from being bright yellow to colorless. So you want to address your observations like before, during, after the experiment, and not just say, oh, well, it looks like this. Okay, describe throughout. Did it change a color? Did something happen? And even if it's not color change, it could be like maybe you saw bubbles being given off. Because bubbles, if you're not heating something up, bubbles are an indication that there's a chemical reaction happening. And so those are some things for qualitative. Within the tables, as I mentioned several times already, you got to have units and uncertainty in the column heading. Do not put them in every box, in every cell, okay? Significant digits, sig figs, or significant figures, in the data and the uncertainty, they must be consistent, okay? And you have to use the same number of decimal places for your raw data. So what that means, I'm gonna go down here. Look here, the uncertainty right here, plus or minus 0.01, that goes to the 100th place. All of my measurements go to the 100th place. So they all match, okay? So they have to match the uncertainty and they all have to be the same number of decimal places. So even this last one, even though it was 1.10, I did not write 1.1. I wrote 1.10. That's because of these bullets right here, that you have to use the same significant digits and the same number of decimal places. So same thing here, 2 decimals, everything's got 2 decimals, they're all matching in precision, okay? Um, and then it says always process and show your results mathematically in some way. So use averages, calculations with formulas to process your results. You only need to show the work for like one sample calculation, okay? You don't have to show every single calculation. You could just do one example for each type of calculation you do, but you do need to do all of them and then present them in your table, okay? And so here it's showing you like how they average the numbers in like the last column. And then this was their average. And then uncertainties, we're going to get to that later. But you also do need to do your uncertainties. Make sure you have proper number of sig figs in your calculated quantities, okay? So make sure that you're keeping that. Descriptions of what you're calculating and why. So like why are you calculating it? What are you calculating and why are you calculating it? If applicable, plot your data in a graph. You won't always have to do that, but it is good to do that if you're able, because that's a good visual thing. If you do a graph, there are some specific things you need to have on it that we're going to talk about next week. You have to have like these things called error bars. You have to have labels and different things like that. Make sure that you're considering the uncertainties and errors. So when you process your data, you have to do that propagation and propagate the uncertainties in percentage form usually, because usually you're multiplying and dividing. Tables and graphs, they should all have headings and titles. You shouldn't just have a random table with no title or a random calculation. Each thing you should tell me at the top, what is this? What is this data? What is this table for? What is this graph for? What is this calculation for? Graphs should have appropriate scales. So, if you're using a graph and your line only takes up this much space, that's not scaled good. Or if you're doing a graph and it's like really big and it's going off the graph, that's not good either. Labeled axes with units and uncertainties. So like if you're doing a graph and you want to tell me like you're measuring mass down here, then you want to tell me that it's grams and it's actually, let me rewrite that. So you want to say it's mass and you want to say it's, you want to give me the uncertainty and the units. So you want to say it's like plus or minus maybe 0.01 grams, whatever the uncertainty is. So on your axes where your labels, you want to tell me what you're measuring and then also the uncertainty in the units. You want to accurately plot your data points and you want to do a best fit line or curve. You do not do a scatter graph and you do not connect the lines like points like dot to dot, okay? Uncertainty bars were significant, and if you do need to do a graph, we can talk about that because you can use Excel to do that. It's really easy. Once you learn how to do it, there's tutorial videos and stuff. Maximum and minimum gradients, determine the uncertainty in the best fit line, present all the stages of your data processing. So the final result can be understood easily. So this is what I'm talking about, that you don't have to show me every calculation, but you need to show me one example for each part or each stage so that somebody can follow it, because I'm not the only one that's going to be grading it. This is also going to get sent off to somebody in a different state or even a different country, and they need to be able to follow your process, okay? So that's what this last couple bullets are meaning. Make sure that somebody else could follow it and understand what you're doing. And then again, be really careful with decimal places and sig figs, okay? All right, the last section before like communication is evaluation. So this is how you provide evidence of evaluation and results with regard to the research question and the accepted scientific context. So you're going to refer to your research question, refer to your data, and also refer to your background research, your scientific context. So on this one, the first one here says a detailed conclusion is described and justified, which is entirely relevant to the research question and fully supported by the data presented. Okay, and then there's obviously other ones like you do relevant comparison to the accepted scientific context. So that goes with your background research. So if you do something related to titrations, you have done background research for acids and solutions that you can tie in to your data and your conclusion. If you're doing calorimetry, then maybe you're looking up the energy or the calories that are in certain foods that you're using to refer to for your calorimetry lab, okay? So tips here for the conclusion. If there's any trends in your data, so this is why it's good to do a graph so you can see if there's trends, you should refer like this increased or this decreased, or maybe it went up and then it went back down, okay, whatever happens. You should compare to some sort of published data. That's your accepted value, or your theoretical information. So that's where your background research comes in, that you've done some data, you've done some research, and you found something similar to yours that you can compare it to. Weaknesses. Weaknesses in the experimental method, you should identify and discuss them. So if you had things that caused you to have systematic error, what weaknesses could you fix in your lab? Make sure you identify error as random or systematic. So don't just say I have error. You need to tell me, is it random error or is it systematic error? Okay. And then if it's random and systematic, how can you improve it? So improvements and extensions. So how could you fix what's wrong with it based on your error? And then how could you extend it? And how could you maybe do something different related to what you're doing, but maybe a slightly different extension? Use your scientific knowledge to explain your conclusion. Make sure you always refer to the evidence in your data, in your results, okay? And then if you're measuring something that has a known and accepted value, you want to compare to the literature value, and you want to calculate the percent error, okay? And then based on your percent error, you can discuss it. If it's negative, that means your experimental value is lower. If it's positive, your experimental value is higher, and discuss whether you encountered these errors. And then evaluation is identify random and systematic errors, state any assumptions. Like when we do calorimetry, we assume that solutions are going to have the same density and specific heat as water, okay? There are some other things that we assume sometimes depending on what you choose to do. And then how can you remove the random systematic error and reduce them? Suggestions for fixing things, okay? Don't list irrelevant sections. So don't just list something because you're trying to come up with something to put in there, okay? And a good way is to structure it like this. So like here is a weakness, and then here's improvement. Because if you identify it as a weakness, you need to suggest an improvement. Don't tell me it's a weakness and then not tell me how you could improve it, okay? And then the very last is communication. So this is kind of the whole outcome of the report. And so basically it's things like I mentioned earlier where, the presentation's clear, it's easy to follow. If there are any errors, they don't like keep me from understanding it. It's well structured and clear. It's relevant and concise. You use terms correctly. So you're not using, terms for like calorimetry or titations wrong. You're not using chemistry information incorrectly. This includes things like labeling. So graphs should be labeled, tables should have headings, sources should be cited in appropriate format. And we're going to talk about the format later on next week. It's similar to what you guys do in English papers. All measurements have units and are recorded with sig figs. Anything that's been referenced using an appropriate system, okay? So all that kind of stuff. And then the rest of this just goes into talking about general lab safety. So if you need safety stuff to incorporate in, you can do that. Then it has a recap of errors and uncertainties like we covered in topic 11. So if you are having trouble understanding like random versus systematic and how to propagate your error and do percent error and all that stuff, you can come down here and there's kind of a recap of that stuff from topic 11, and specifically how are some things that you could what are some things you might see in labs. A recap of significant figures and decimal places for recording your data is on here. Here's the example for calculating the uncertainties, so propagating that error. There's one with addition, subtraction, and there's one with multiplying and dividing, so you can see the difference for how you need to present it and how you should do it. I will say there is a typo right here because these are both 0.5. This one, it should be 1.0 when they're added together. For some reason, it's a typo. So it should be 1.0 degrees Celsius when you add those two together. And then at the very end here, it has common lab equipment and what their precision and uncertainties are. So like if you're measuring mass and you're using electronic balance, it's always going to be plus or minus 0.01 grams usually. If you're using, beakers, graduated cylinders, then it's usually plus or minus half of whatever the smallest scale division is. A lot of these other ones that are more specific, they have their own listed over here. A regular alcohol thermometer is this. If you have a digital thermometer, that's going to be a little bit more precise. I think it'll usually be plus or minus maybe 0.1, 0.05, something like that. So it depends there. And obviously there's other stuff, so you can add stuff on here if you need to add something for what you're using. We're not going to be using any of those probes. And then here's just a whole worked out example. So this is somebody that they did an enthalpy experiment, and it's going to show you step by step sort of how they worked it out and how they propagated the error. And then the very end here, it just has a table of units. If you are not familiar with some of the units that you would use for some of the measurements, it has the units here and some conversion factors for volume. And then again, there's more units. And then it also has the prefixes for metric units. So like if you have like centimeters or milliliters or things like that, that's at the very end.